Yeah, we'll have some multiple choice questions that will some be from the study guide, some from other materials that are in the slides, and then the short essay questions will be directly from the study guide. Um, Study guide is like last time, it's more to encourage you to broadly study, but also to think more about the conceptual stuff that we're trying to learn in this course. Any other questions? Today we'll talk about object recognition. If you look at this slide, I think it says something about attention too, but I'm not going to start a new topic today. We're just going to try to finish object perception, and then the exam will be up to whatever we cover today. And then after the exam, the following week will start on the attention topic. You've all turned in or the ones that wanted to improve your grade, you turned in your assignment one resubmission, and hopefully I think you'll get a grade for that in the next week or so. But it just to improve your grade, so it's not something that um you're waiting for or anything like that. So yeah, just to study for the exam this week and come prepared. So today we're going to talk about object recognition. Now, this is a lot related to this stuff that we've been talking about, especially last week, and also to everything else we talked about in this course. We were kind of building up to this lecture in a way that so we started talking about single neurons. We talked about the physiology of a single neuron, what a single neuron does, how does it emit an action potential, how do neurons communicate, how does a pattern of activity in one layer can lead to another pattern of activity, another layer, and then another layer, and then another layer, right? We talked about, you've done these practices in order to get comfortable with the fact that one pattern of activity, depending on how these neurons are connected to the neurons in the next layer, going to cause a certain excitatory activation in some, it's going to reduce the activation in some other neurons. They have another effect. So it's a cascade of effect. And we talked about in the context of pathways. Well, some of you would have asked, well, what does it mean if this neuron is responding this way, or if it's being inhibited? Like, what is the perceptual effect of that? And I think today we try to bring some of that into it. Like, what is the perceptual effect? What is the eventual outcome of going through this cascade of processing? And object recognition or generally object perception is an important cognitive ability, right? Everything we do in the world, it involves us interacting with objects, right? We recognize objects, we interact with them, we manipulate objects around us, we use certain objects as tools for other objects. So objects are really the units that we interact with the world and we perceive the world. But objects are not properties of the world, meaning that they don't exist necessarily in the world. It's just that we perceive certain set of physical entity to be an object and others to be another object. But our ability to recognize them, to understand, well, these chairs are the same objects. These people are kind of similar objects. Those chairs are a different object. So this categorization task is very fundamental to anything that we do. But this is a very difficult task in many ways because the stimulus that we get on our photoreceptors are really ambiguous, right? We talked about very simple photoreceptor patterns, but these are like pixels of an image. You could have images of the same category that are so fundamentally different in their pixel values, right? So here is one example. You're looking at images of one category, right? It's remarkable. If you don't know what this category is, it by itself is remarkable that we could look at these images and recognize they're from the same category. right? Because they're so different looking. So this image is very, very different looking than this one, and then this one, and then, this one or that one. These are very different images, but we're able to kind of see the similarities between them. Now, if you can see the similarities between them, and if our brain is able to do that, and if I told you every act of perception is some neural activity, what does that tell you if I look at all these images and I have the same perception from them? That perception is a pattern of neural activity in my brain, right? When I look at this image, that causes a pattern of neural activity in my brain. If I look at this image, that causes a pattern of neural activity in my, and if I say these are the same category, what could that mean for that pattern of activity? If my perception of two objects in some way is the same, right? I know that these are two different images, but at some level, I say these two are similar. Convergence? No, okay, good. Yeah. Mm-hmm. Yeah. That's a good point. Yeah. There is some top-down processing, but in this case, you could do this potentially with feed forward. Yeah. Okay, that's yeah, yeah. That's it's probably happening in the same part of the brain. What part of the brain would that be? For object recognition, which part of the brain would that be? Which part of the brain we talked about last time for object recognition? Which part of damage to which part of the brain causes damage to object recognition? What's that? No, no, no. What? So if I talk about visual form agnosia, which part of the damage to which part of the brain causes me to not recognize? Temporal. What is the area that we talked about? Infratemporal, right? Let's say the area IT. So there's an area that we talked about, and this is part of which pathway? The ventral pathway. Okay, so this is part of the ventral pathway. And the area that we talked about is the area IT. So yes, these visual representations are being processed there. But what does it mean if I perceive these two to be the same animal for that representation, for that neural activity? This means if I say my perception of this object is some pattern of activity, and I perceive the two objects to be the same, it means what for the neural activity? What's that? What's that? Same pattern. That's the answer I'm looking for. If my perception of two objects is the same, there must be somewhere in my brain that these two are producing the same pattern of neural activity, right? If you accept that all of my perception is reduced to neural activity, if you accept that reduction, then it means that if I'm seeing two things and they're similar, there must be some neural activity that happens similarly, right? If I'm looking at this image, there's a pattern of neural activity in my IT that is similar to the pattern of neural activity when I look at this image. Does that make sense? Because that pattern of neural activity is signaling the identity of the object I'm looking at. And similar representation, and representation would be another word for that pattern of activity. That similar representation should be there for me to have the same perception. Does that make sense? But these two have nothing in common in pixel values, right? If I'm looking at this image and I'm looking at this image, if I take every pixel here and convert with every pixel here, is there any similarity, you think? Well, no, this area is brown. This area on this side is brown. So the pixel values would not match. What does it mean for the retina if the pixel values don't match? So the activation of photoreceptors would be different, because we talked about the retina is very close to the pixel values. If you're looking at an image on a screen, and if you're looking at the world, it depends on the activity there, right? So what we talk about today is the fact that the visual input that you get on the retina is very ambiguous. And that's what ambiguous means. It means that you could get the same category, but what you get on your retina is very different. So which one is a koala? If this category is koala, which one of these would be koala? The one on the left, right? This is the fact that we could do this is remarkable, because when you look at these variability that it is in here, and then you look at these two images, the image on the right has a lot in common with all these figures that you see here. It's in a green background. It's hanging from a tree or something. It has the same kind of skin texture, somewhat similar, facial features. But this is the difficulty of object recognition. The stimulus it's only invariant to the properties of this stimulus, right? You get very different perception between the two. You may get very similar perception on the retina level, but you get very different perception when you go higher level.
What if someone had never seen whatever animal that is upright before, and they couldn't make that prediction, and they was close enough that those pathways, because part of this has to do with probably the memory of having seen a koala at some point, right? It's predictive processing, okay, it's going down the same pathways. But someone's never seen a not koala, let's call it, how would they think it was one?
Sometimes, yes, if you're close enough, like I know examples, like let's say there is an Australian sheep, species that you've never seen. And if I show that, you may think it's a sheep. But for someone else, you may think that it's not exactly a sheep, but you would not know it's a whole different category. In this case, again, we are so good at this that we could form clusters even without having access to what the other thing is. In this case, even if you don't know what a sloth is, if you've never seen this, you still could kind of say, I don't know if it's the same category. Because you look at, and again, we're very good at abstracting across samples. If you give people 5 samples of a new category, they kind of form a new category. And that's what makes us very good at life, abstractions, that we can actually look at multiple situations and be like, okay, this is what's similar. And when you're in a new situation and you want to make a decision, you're like, okay, these are the things that are similar, that are familiar to me, and I'm going to make my decision based on that. So we are very good at this. We look at just these examples, and even if you've never seen a koala or seen a sloth, you could kind of tell me that this is part of that category, and the one on the right is not part of that category. So we're very good at forming boundaries around categories. Um, but if it's really close, well, people would mistake. So what makes these representation to be ambiguous? Well, a lot of time, visual input and objects are hidden or blurred, right? So in the example that I'm showing you here, in any of these images, you don't really see the entirety of the object. So here, maybe you see the entirety of the object, but in this case, you only see, it's like a baby koala and you see only the head and part of the hand. In other examples, you see part of the objects. So that makes object recognition very difficult, right? So if we are in an environment, a lot of time objects are occluded. They're occluding one another. So we get a small, like when you're driving or you're crossing the road, a lot of time you just see parts of the car or parts of the person. You don't see the whole thing, but we still are able to recognize them. Occlusions remove parts and features and make it very difficult for us to say, okay, is that the whole object? But again, we're very good at this. Um, but a lot of time you realize things are difficult when you try to make a computer, like when you write, an artificial system to do it. To us, it comes easy. Well, this is these are scissors, and there's a cognitive psychology book there. Even though a majority of it is occluded, we kind of could read it and kind of make inference about it. So that's something that we're very good at. Another thing is, again, a lot of time we look at objects not clearly, right? It's either raining and you're in a car and you're looking at through, dirty glasses or something, and you don't get to see objects very clearly. And that makes object recognition very difficult. But I imagine you all recognize these individuals, like very, very topical, Who is this? So that's what I was saying. Is there a generational gap here between people who can recognize things? Who is this? Which is King Charles now.
There's a generational.
Yeah. This is Bill Clinton, and this is Anna, and Nixon. So this is not an easy task to recognize faces among millions of faces when you get such a blurry input. And again, it shows how good we are at this. Objects look very different from different viewpoints. A simple example, you're looking at a chair from different angles, it's going to look very different. And I say look very different, I mean pixel values. If you look at the examples that we had here, like this viewpoint is very, very different than this viewpoint. You just get very different features. Again, because we're good at it, we don't see it, but in the pixel value, these two have nothing in common. But we're still able to recognize them. So again, this is another thing that makes object recognition very difficult. Another thing is that objects really change appearance when the lighting changes. Either the intensity of lighting changes or the direction of lighting changes. And I have a demo here for you all. So considering those three factors that I mentioned, the fact that objects change under blur or occlusion, the fact that different viewpoints change, like visual input, and the fact that different lighting change the input. How many different identities are here? How many people think there are 5 different identities?
5 separate people making different faces.
Yeah. How many people think it's only one? How many people think it's 3 different people?
It's more than one.
It's more than one. How about two? Two different people, no? Four.
Four. I think it's four.
This is only two different people.
Oh.
So, so the thing is, another factor, right? And you saw that in the koala example too. Well, people look different at different stages of life. And these are over a certain period of time. It could be facial hair, it could be your hair changes. Like a lot of things change when it comes to recognizing people. Um, but you could see all of these people that are in the green box, like images that are in the green boxes are the same person, but at different time, a point. So, and again, lighting changes so much, you know, when the light is shining from the side or from the front, or when the picture is blurry and the facial hair is gone, all of these things change the identity, like change the physical appearance. It changes. And again, this is, I want you to internalize the problem of what is the challenge of object recognition? That what you get on your retina is very different every time. Every time you look at someone from a different angle, different lighting, you see parts of their faces, or that they're, blurry, occluded. In all of those conditions, object recognition becomes more and more challenging. But we're able to go from an ambiguous retinal image to an identity that is invariant to all those changes. And we briefly talked about the concept of invariance last time. What was it? We talked about invariance in the context of IT. What is invariance in the context of IT? What does invariance mean? If a cell is invariant to size, what does that mean? What's that? It's not sensitive. So this is the opposite of coding for something. When a cell, let's say if I say this cell codes for the direction of motion, what would that mean? Sensitive. It means what for its firing rate? It means that if the object moving in this direction or in the opposite direction, the cell is going to respond differently. So what does it mean when it's invariant? If I say this cell is invariant to the direction of motion, it means what? So the response doesn't change when the direction of motion changes. It's invariant, right? It doesn't change with that underlying factor changes. If a cell is invariant to a scale, it means that that scale doesn't change the response. Changing that scale doesn't change the response of the cell. So the problem of object recognition is going from ambiguous input on the retina to invariant representation in the higher visual areas. Yes. It makes them more photogenic. Yeah, like is there something, but or is it like, yeah, I don't know. Is it like it could be like the person too, or like? I think some people. I don't know if I'm sure somebody has done a study on that, trying to, you know, boil it down to certain factors that make someone look good from different angles. Um, what's that? Camera lens, you think so? Yeah. Um, you could probably take a better picture from anyone if you try. Okay, so these are the three factors that we mentioned that makes the stimulus on photoreceptor ambiguous. They could be really degraded. Degraded meaning that, well, you get parts of the objects or it's blurred or something. They could look really different from different viewpoints. And with the change in lighting and illumination, objects could look really different. So a lot of the stuff that we talked about brings us to this point. And this is one way to think of the visual processing, um, which is that a lot of visual processing is feed forward. That the visual input starts from the eye, it goes to the LGN, it goes to the primary visual cortex, and then from there it propagates forward in the brain. And the process that happens across this hierarchy gives you certain selectivity. So if this is this dog, you're looking at this dog, right? And we saw examples of this earlier too, but this is putting it, across the entire hierarchy. So if you have a photo of the dog, as you have here, the V1, the primary visual areas, are processing this visual input in parallel. What does that mean when I say the primary visual cortex is processing this visual input in parallel? All at the same time. Why? Well, because we talked about the retina topic mapping. When you look at the visual input, when you look at the visual world, this visual input is projected onto your retina. It goes to your LGN, it goes to your primary visual cortex. And the primary visual cortex has these feature detectors, right? Orientation sensitive cells that are tiling the entire visual cortex. So when they're tiling the entire visual cortex, it means that at every location in this space, there is a cell that has a receptive field, right? There is a cell that has a receptive field here. There's a cell that has a receptive field here. Does that make sense to everyone? That there are cells that are tiling this entire visual input. And they have receptive field on every part of this dot. Now, the receptive field of, supposedly, as we talked about it, if the receptive field of this cell is here, the receptive field of the cell next to it would be where? It would be somewhere nearby. That's what the retinotopic mapping is. So you have all these cells in the primary visual cortex, and when the image projects into that, it kind of gets processed. So based on the response of those cells, you know where all the edges are because they're detecting all these, boundaries. So what happens at the next level? Well, you have these, and these would be simple cells, right? Then we talked about it, that simple cells are really orientation selective, so they respond to certain orientation. The simple cells could together have different selectivity, right? If you're detecting edges, then a cell that is connected to all these edge detectors could potentially detect a corner. And the cells that are detecting corner could potentially feed into another cell that detects boundaries and shapes. And those cells could fill into other cells, and those cells could detect maybe a face, right? So this sorts of feed forward abstraction of selectivity happens in the visual cortex, right? We talked about this in the context of the ventral pathway last week, right? We said, okay, if some cells are detecting these circles, those cells after that could combine that response, detect edges. Cells after that could combine that response, detecting no corners, and so on. Does that make sense to everyone? Does that make sense how at the end of all of this, you may get a cell that is responsive to faces and not like certain kind of faces, and it doesn't respond to anything else, just faces. Because, it's connected, it's getting its input from cells that are detecting maybe eyes and lips. And those that are detect eyes are getting certain kind of input.
On the right-hand part of the slide, S1, C1, S2, they're layers, but what are the?
So these are simple, complex, simple, complex, simple, complex. This is a certain organization of people propose that you have this sort of simple, complex processing. And the simple one are more fundamental features, and then complex is like a combination. But on the left side, we have these brain areas that we talk about. A lot of these edge detection and feature processing would happen in the primary visual cortex, as we talked about. Then, so primary visual cortex is also called the striate. So when you say extra striate is the area outside of primary visual cortex, extra striate, visual area. And then this is the IT, the inferotemporal area that we talked about, where it's in the temporal lobe. And it's called infratemporal cortex because it's infra in the temporal lobe. And there are two areas that they discuss. Again, these mappings are not clear-cut mappings. These are just more so of a, general mapping. That's what they mean to communicate. Now, why do people think this is how object recognition happens? Partly because object recognition is fast. That when you flash images with even 100 milliseconds, people can recognize them. So if you flash 100, and again, I wish I had a demo, I could look for it later. But people could actually recognize objects every 100 milliseconds. But there are delays between these areas, right? The synapses, because the action potentials have to travel across an axon, it takes time. It takes 5 to 10 milliseconds to go from one layer to another. So if you have visual input on the retina, it takes 5, 10 milliseconds for it to reach the LGN. It takes another 5, 10 milliseconds to reach the primary visual area. Another 5, 10, another 5, 10. So if you add up all these delays that you get from the visual input to IT, it's going to be around 100 milliseconds. So it's like the system has no time to have these recurrent and feedback connections. Everything, all the processes are feed forward at this point. Does that make sense? It's just the delays that it takes to go from one layer to another layer that are accounted for in this 100 milliseconds. So this is the feed-forward theory of object recognition. That processing happens along a hierarchy. It's predominantly feed-forward. That one pattern of activation leads to another pattern at the next layer, until you reach area IT. And there you have cells that are category selective, that they don't respond to just, they're invariant to the size and the scale and the color of the object maybe, but they vary along category representation. The book has a different version of this figure, so I wanted to include that here too, where you could see, and again, this goes from top to bottom. So top being the more simple features, and these simple features become conjunctional features, and those conjunctional features become more conjunctional features, and then become an object. Now, a cell can only respond, again, this is a claim about hierarchy. The cell could only respond to a certain shape if it getting its input from cells that are responding to its component. Does that make sense? This is very fundamental, right? A cell cannot detect the shape unless some other cells have detected all the corners and are communicating to this cell with their action potential that we detected some corners, and this cell, okay, now that I see, these thousands of cells have detected corners, I'm going to say, actually, there is a box there. Otherwise, there wouldn't be 4 corners. That's why you need hierarchy. At each stage, you have to, build on it, build on your feature process. Yeah? So is that really the case in the visual cortex? So let's look at an experiment. The experiment that I showed you earlier in the course, right, with the video and Hubel and Wiesel, and I said they won a Nobel Prize for it. That experiment is from the 50s and the 60s. So they're 60 some year old experiments. Well, what was the fundamentals of that experiment? Well, you're recording from a certain cell, you're changing your visual input, and you're trying to find that visual input that maximally activates the cell. Right? That's the fundamental aspect of that experiment. And how do you change it? Well, there are two people spending months on it, changing, playing around with all the variations of light, moving them around with different sizes, different orientation, different speed, different direction of motion. They try all the possibilities to do that. Now, fast forward 60 some years, and we're still there in a lot of ways. This experiment is from a couple of years ago. They said, why don't we automatize the process of experimentation of what pattern activates the cell most? So what they're doing is that they're recording from a cell, let's say in the area IT. They're showing a bunch of images, synthetic images, and recording their responses. Now, if you have a bunch of synthetic images and you record all the responses, you could see which one of them maximally activated this cell. You could see which one of them had the highest activation. You could look at the firing rate. Then you could combine them based on that. You say, okay, the one that has the highest activation, I'm going to weight this more, and then I'm going to add up the other one to it. You create new stimuli, and then you use genetic algorithm and some optimization, but don't worry about it. You could create a new set of input, show them to the monkey, record again, then repeat this process. So what you're doing the same Hubel and Wiesel experiment, pretty much. But instead of an experimenter sitting there playing around, what you're doing is that you're really playing around with the visual input in an automatized way. And when you do that, you find these funky patterns. Like this shows the evolution. Like this is recording from a single cell in IT, for example. And let me make this bigger. So what they're doing, as I said, they start from a certain pattern, and then based on the activation to these patterns, you want to combine them, make new stimuli. Then you want to see that activation, combine them, new stimuli. And over time, you could optimize it. And this cell, I'm going to stop it at the end this time. So this cell happens to really like this visual pattern. Now, at this point, because this is a higher visual area, the pattern would not be a simple oriented bar or something. You would expect it to be a complex visual pattern. And in this case, it looks like maybe a monkey face and body, right? So a cell in the higher visual area would have a selectivity that is more toward objects. Does that make sense? How this is basically that same experiment. It's just done with more modern tools, where you're trying to really, so again, the cognitive neuroscience is about finding what a cell does a lot of time. And what a cell does depends on where it is, what kind of input it takes, right? And that's, a lot of the research project pretty much. You want to find out what a cell does. And in the higher visual area, this is what it does. Now, I can tell that this cell detects monkey faces. That's one way to put it. It means that what? It means that it really fires strong. It has a high firing rate when it sees monkey faces. Okay? Now, let's look at an artificial neural network. Now, this is a network. I gave you very simple examples in the first module, but this is a much bigger network, imagine. It's like almost, as big as a brain area, for example. It has many units that goes from the pixel image to an identity that they try to predict. But what you can do with an artificial neural network is the same experiment you did here. You could look at a unit in this network and basically play around with your visual input and see which visual input maximally activates this unit. Instead of doing it in a monkey, you're doing it in an artificial system that is trained for recognition. And what they see is something like this, that across these layers, neurons that are units in this artificial system that are closer to the visual input, that are in the earlier visual areas, are responding to edges. And units, as you go higher up this hierarchy, they become more selective to face and house and better concepts. Does that make sense? What this hierarchy of selectivity means? That you become more object selective. That in this area, units are responding to things that look like objects rather than to just visual patterns that are meaningless.
Are we seeing like a film of an area of the brain that has lit up and that looks like a face? I mean,
no, we're looking, this is from an artificial neural network. So this is not the brain. What we're looking at is that what maximally activates this unit in this network at a certain level. So as I was saying, the ANN, artificial neural networks that I gave you are just a single layer. You had input and output. But imagine this has many more layers. And it gets visual input and it predicts the category of that visual input. Now you could look at different units in this artificial neural network. It's not a brain, and see what is selective to. And that way you could tell if it's actually capturing the same hierarchy as the brain. That's the idea. So in an artificial neural network, you could find units in the higher layers that are selective for categories. That's what I'm trying to show. Similarly to what you saw here. Like these people have found cells in the IT that are selective to these categories, but you could see the same thing in an artificial neural network that is trained for object classification. But I just want you to see this hierarchy and imagine something like this potentially happening in the brain too. That you go from V1 to V2 to V4 to posterior and anterior IT, you get similar hierarchy of selectivity. So that's what I want you to take away from this. That different layers in the brain, the cells are being responsive to different levels of hierarchy, in different levels of hierarchy to different levels of abstraction. So having told you this, people think, okay, what if I take this idea of selectivity to an extreme, right? If you're able to, across multiple layers, build such a specific selectivity, could it be that there is a single cell or a bunch of cells that only fire when I see my grandmother, right? Like that when I see my grandmother or I hear her voice when she calls me, or when I meet her, or I think about her, in all of those instances, there are a few cells in my brain that are responding to only that, right? So this is extreme level of selectivity, that those cells always are signaling my grandmother, right? And this idea is called grandmother cell. And sometimes it's called Jennifer Aniston cell because there is a study done. Again, this is one of those examples that I gave you that where you could actually run a neurophysiology experiment with human subjects. And what is the example that I gave you? What is the condition? What is the situation where you could run neurophysiology experiment? No, neurophysiology experiment in humans. No, so what can? No. Neuro ARP is a different method. Neurophysiology, single cell neurophysiology with humans. And under what circumstances can you do that?
You're talking about the eye thing?
No. I'm talking about under what circumstances can you actually put an electrode in human brain? Seizure patient, right? So this is a case of that. These are seizure patients. They're in the hospital and they're bored, but they have electrodes in their head because you have to constantly monitor them. Now, if you're a vision scientist and you get lucky and you get a seizure patient that is being monitored in a certain area of the brain, you could do vision experiments with them, right? So in this case, they did those experiments and they showed different images of different celebrities and people to this patient. Now, this is two different ways of showing firing rate, right? I showed you both of them. This is a raster plot where it shows the individual firing of the cell. You see all of this? Each of these and each row is a trial. You don't do a single trial, you do multiple trials. So in this case, you do multiple trials, each row is a trial, and each dot, each blue dot is a what? An action potential. Each dot here is an action potential. Each row is one trial. So you do it again and again and again at some point, and each dot is an action potential. Now you could summarize this in a histogram. Basically count how many action potential was in each of these windows across multiple presentation of the same image. So here you could see that this cell is responding very strongly to these images and not at all to the other ones. And what do these have in common? What's that? They're all Jennifer Aniston. So the fact that they're all Jennifer Aniston and only these cells, this is just ordered. This is not the order that they were presented. This is the order that they show here. And what they're showing is that this cell seems to be selective to Jennifer Aniston. It doesn't fire as strongly to other faces or places, and they have this like Eiffel Towers and the San Francisco bridge and everything.
Yeah, but it's not compared to other pictures that look like big stars.
So I mean, these pictures, like you have examples here of women, meaning that. So what is the fundamental problem that you're pointing out? Is that can you really test everything, right? So the fundamental issue is that if you want to claim that there are cells that only respond to Jennifer Aniston, it means that you've tested everything, right? And that's impossible. You can't test everything. So that's the fundamental issue with that. But as an experimenter, you try to do the best you can, which is you say, okay, can I control for all the other things? Can I say, well, what if it responds to women? What if it responds to celebrity women? What if it responds to fake blondes? What if it responds to someone who was on this specific show? Or what if, so all those possibilities you want to consider, but you can't. So that's the thing. But at the end, you cannot really consider all of those possibilities. So there's always a limit to how much you can really understand from an experiment like this. But it's interesting in some ways that among all the things that they tried, it seems to be that, oh, this cell is only responding to Jennifer Aniston, for example. For this reason, for this experiment, it's also been called Jennifer Aniston's Nora.
I mean, like, did they show, for example, photos of Jennifer Aniston when she was very young?
Yeah. So that's what I mean. So they did, yeah. So they have photos across different. Again, that's the thing. That's the problem, right? How many are you testing? What range you're testing, and all those sorts of things. But let's assume that they did all the obvious ones. Let's assume. But you can also check out this paper. So uh, this is what a grandmother cell is. The concept is that if you're building this selectivity across many layers, um, then you you can definitely get to a point eventually that you're so selective that you're only responding to one identity, like across this hierarchy. And that's how you encode maybe concepts. That's how when you listen um, to someone talking about this person, or when you see a video of them, or when you hear their voice, they all bring the same perception. Maybe that's why, but that's a theory.
Going back to the grandmother cell concept, is only that like, is it a cell that gets so highly specialized that sort of becomes coded after, like, say, a baby that doesn't know their grandmother and or doesn't see their grandmother until they're 7 years old? So do they have that type of like template for that specialized cell, or does it sort of coding change as you grow older or recognize more faces? Like,
Yes, so part of it is that it's a learning process where because you see certain faces a lot, you develop very specialized uh, machinery for them because you see them so many times and for so long that they become very specific. Um, but we're also good at recognizing people that we don't see that many times, right? And you could recognize them later in life based on the certain features, and that could be a more shared thing. So there could be somewhere in between, right? There could be both very specialized machinery for people that you have intimate, familiarity with, and people that you don't. Um, but it's a you got to take this with a grain of salt in terms of how likely it is that all these concepts that we know could potentially be having their own little, patch of cells that are coding them. Let's see. So let's take a 10-minute break, and then we'll come back and we'll talk about an alternative. Cool. All right, so I first wanted to show you because I said I'll show you a little later. So this is a rapid serial visual presentation paradigm, which is basically you rapidly are showing images to people. And this is just to show you how quick object recognition is. Um, your task is to reboot whether Buzz Lightyear, which I imagine is a Toy Story personality, is present in each sequence. If you see Buzz, press the right arrow, but you could say Buzz if you see him. If you don't see Buzz, or whenever you see Buzz, say Buzz, I guess. Um, okay. Okay.
but
Oh, there was a Buzz.
that's not Buzz.
That's not Buzz?
That's not Buzz.
All right.
That's a different representation.
Okay, that's not Buzz. All right, let's see.
Transformer.
So no Buzz so far? All right. Yes. Let's see if, oh yeah, okay, that's where it is. So this is a demo to show you that these images are flashing 100 milliseconds after another, but you're able to detect a complex object among, all these things that are going on. So that tells you something about vision. That's how fast it is, and it shows you some evidence for this idea of feed-forward processing, right? There is only 100 millisecond time for the visual input to go from your eyes to your IT. If you are able to recognize something in 100 milliseconds, then it means that all of it is feed-forward, that there's no time for things to come back and modify your representation. Okay, so uh, so now we talked about the grandmother cell idea, and the type of coding that grandmother cell is this sort of what we call a specificity code, meaning that you have a neuron, like imagine these are three neurons, 1, 2, 3, and they could have different firing rates. And this is, let's say, how many action potential in 1 second. And let's say this is, I don't know, 10 action potential in 1 second. You could have a system that responds when it sees this person, only cell one responds. So this is the grandmother cell for Bill, this is the grandmother cell for Samantha, and this is the grandmother cell for Roger. Basically a cell that only responds to Rogers and does not respond to anything else. And that's the idea. That's how you would expect the system to behave. And it's a specificity code. Your code is a specific to individuals. Alternatively, you could have something that we call distributed code, where the identity is not specific to a cell, but rather to the ratio of activation among all cells. So the representation, as if you like, is distributed across cells. So this distribution of activation codes Bill, this distribution codes the second person, and this distribution codes the third person. So it's a distributed coding of the same concept.
So does that mean it's distributed code for face space, and then specificity is Bill face and Matt face and Roger face? I don't think I understand what.
So let's say you have a system that is simplified and only has three neurons. 1, 2, 3. That's how simple it is. And you could think of it, if I come up with an example for it, you could have, you have three friends, and you could identify them by, okay, this person is, cell one, or maybe a certain color or something, and you assign a color to a person, right? What is the problem with that? What if you have too many people to code for? You need what? You need more colors, for example, for them, right? So if I say, okay, I have a phone book and I color code them, and for every person I pick a new color, then there is a problem. I'm gonna run out of colors very soon. Alternatively, I could code every person as a ratio of colors, right? So I could be like, this person is a little green, a lot red, and maybe a moderate yellow. I could code them that way. I could code another person as a little bit more red, but the same amount of green and yellow. I could code another person with a different ratio of those colors. What does the other code gives you? A lot more ways to code different individuals, right? And it's distributed. So the representation of one person, the way you identify one person, is not by a single color, but by a ratio of a combination of a lot of colors. Does that make sense to everyone? The difference between a specificity and this sort of distributed coding. But this is a very simple system with three cells. And in my example, with three different colors. You have three colors, and you were trying to assign them to different people. so you could recognize them again, and you run out of stuff. Another problem is that if you lose a cell, what happens? If you lose the cell number 2, well, in this case, you probably lose your memory of Samantha because you have no way to represent Samantha. But if you lose cell 2 down here, you still can recognize Samantha by the fact that cell 3 is more active than cell 1. A little bit, right? So cell 3, if cell 3 is twice as active as cell 1, you know it's Samantha, because if it was Roger, cell 3 would be 3 times as more active as cell 1. Do you see what I mean? So if you lose a cell, you still, based on this distributed code, could retrieve, represent a person that you couldn't do with a specificity code. And those are the 2 examples that those are the 2 good things about distributed code versus the specificity code. One, well, if you need to represent more people, you would need more cells in a specificity code. You need more grandmother cells for them that are only active for them and nothing else. On the other hand, when you lose a cell, you have a much more graceful degradation of memory and abilities when you have a distributed code than you have a specificity code. So in a distributed coding, representing a pattern as activity distributed over many neurons or neuronal groups. So if I'm looking at my grandma, or I'm looking at one student here or another student, I'm representing their features as this distribution of many groups of neurons from many different areas that code for color, they code for their identity, they code for their facial features or their facial hair or something like that, and all of those together, the ratio and their activation together represents that person in my head. I'm representing that person in my head using that combination of activation. So a specific ratio of activity among those groups code for different patterns. It is more computationally powerful, right? It could code a lot more patterns. I tried to show you with a simple color example that with a combination of colors, you could code a lot more colors and examples than if you had a color assigned to every person. On the other hand, it has redundancy, right? Because if it's distributed, and in the brain it's distributed across millions of neurons, then losing a few neurons, what is a few neurons? You lose that, you still have a nice distributed coding of that concept. So distributed coding in the brain and in the world, it doesn't have to be in the brain, gives you a more graceful degradation, but also it gives you more computational power. You could code for more information using a distributed coding. So getting these ideas together, and somebody pointed this out earlier, you could have then a combination of these ideas where not only you have distributed coding, but each of those neuronal groups are coding something specific, right? If I have a group of neurons that are coding for a specific hair type or a specific facial features or facial shape or something like that, then all those things together could become a perception of my grandmother or a perception of a person that I know. So it's a combination of ideas usually, right? You have things that are somewhat specific because it has to be specific to some degree, right? If it wasn't, we would never have a memory of a person. If there wasn't something specific to them that activates something in our brain, then we would never have a memory of them. We would not be able to recognize them the next time we see them. So there has to be something specific, but it doesn't have to be on the identity level because, well, how many neurons do I need to code all the identities and all the concepts that I know? that you run into that issue. So it must be somewhere in between, where you have a specific codes for different feature types, and then you combine them in order to recognize. Does that make sense? How these ideas could work together in order for you to have a perception of a face? Okay, so far we talked about this idea of feed-forward process, that you open your eye and the visual input enters your eye and becomes neural activity and just goes through this pathway to the back of the brain and then just basically comes forward through this pathway that we talked about. And they become identity, they become color perception, they become all sorts of things. However, there's more to that, right? It's not everything is that simple. It's our memory of objects plays into it. Our understanding of how things work in the physical world gets into it, right? You perceive things because you have some understanding of physics of the world. You have some understanding of the physics of the light, motion, all those sorts of things. And those things actually play into our perception. Now, a good example of that is this, basically the way that you perceive these. So these things, do you perceive that they're mounds, they're jutting out, or do you perceive them as if they're going in, as if they're, I have another example down there. All right. How do you perceive these things? Do you think these discs are like this or like this? How do you perceive these? Do you feel them as if they're coming out of the screen or they're going into the screen? What's that? Right? So you're kind of perceiving these as convex, right? They're coming out of the screen. They're coming toward you. But why do you perceive them, right? These are on two-dimensional spaces. How about these? With these, you perceive them this way, right? You perceive as if there is an indentation in the screen. Why do we perceive them that way? Well, because we expect the light to always come from the top. That's like our understanding of the physics of the world, because the sun, and people have shown that people in the northern hemisphere expect the light to come from not only from the top, but from the top left. Like they always expect the light, like when kids draw something, the sun is always on the top left or something, right? It's just that's your expectation of the world. And that means something. It means that if the light is always coming from the top, right? If the light is always coming from the top, and you see the light on top of something and not at the bottom, it could only mean that it's coming out, right? That there's no other way to interpret that. But if the light is coming from the top and it's kind of, shady up here, but light down here, it means that it must be going in, right? So the fact that you assume, and this is like a deep perceptual assumption, the fact that you assume the light is coming from the top changes your perception of these disks just based on where they're brighter and when they're dark. Does that make sense? So that's a world knowledge. That's a world knowledge that your knowledge of the physical world, your knowledge of the fact how objects reflect light, that changes your perception. The Bayesian, yes, it has. Where did you hear that term? Yeah, so again, this is outside the scope of this class, but it's about your prior knowledge of the world, like how you bring your prior knowledge of the world into anything that you do, right? Your expectation plays into it. Your expectation that the light is coming from the top, from higher to down, changes your perception of the world. Look at this example. So do you see this pattern in the sand as indentation or as mounds? Mounds? Like these patterns?
Yeah.
Do you perceive them as mounds?
I do.
Interesting. Do you all perceive them as mounds? Wait a second. So how about this one?
You're looking, looks like you're looking at the bottom of a planet like this. They still look.
Wait a second. This one,
I just see it. Now I see this invitation.
right? So these are mounds, right? Unless I'm,
But I only saw it that way until you showed the other.
yeah,
It's the blue dress.
yeah. So, but wait a second. Now that we have the two, between the two, which one of them feel like mounds to you?
To the right.
Interesting. Huh. Okay. So how many of you think the right one is the mound?
Well, we're looking up, you know what I mean? Our light source is going up, so in a way it could be that. That could be why we see it that way. Like it's
I still see
underlit.
them as mounds, and like those are indentations. to
Now
me.
we see it that way.
Okay.
But it's not until you showed the right hand slide that I see the.
Okay. So again, this was meant to show you a real example, real world example of the same concept. Now it's the same figure that is turned upside down. So supposedly there is still, but why if I turn the same figure upside down, because again the physics of the world, you expect the light to come from the top. But in reality, the light is coming from the bottom here because I'm turning the thing upside down, but you don't expect it. So you cannot perceive this as the same thing. You perceive it as a different thing. Here, because again, you expect the light to come from top left. That's what people expect. If you expect the light to come from top left, you expect this area, if it's indented, you expect this top left area to be shades and then this to be light. So that's why it should feel like an indentation. In this case, the light is here and the dark is here. And if the light is coming from this direction, this should feel like it's a mound, it's coming out. Yeah? So by turning a figure upside down, you're changing this physics of the world in some ways, but people's perception changes because their prior is that the physics of the world hasn't changed. Okay, another thing is that, so that's the physics of the world that we use, and it's just so deep in our perception. Even if I tell you, hey, I turned this thing upside down, that doesn't change your perception, right? You still perceive it the way you perceive it. Another thing that we could use when the visual input is blurry is context. So what is this object on this?
Plate of food.
A plate of food, right? You would think the most likely thing is that it's a plate of food. What is this object? It's a car, right? You're recognizing something about the scene, though, right? Here, you're recognizing that thing in the back is a building. That must be a car, right? What else would be on the street? In this case, you're saying, well, that looks like a dinner table, and I see some bottles around it and the people sitting behind it. That must be a dinner plate, right? What else could it be? In reality, these are both the same pattern, right? It's just, you just copy this pattern over here. So the pattern really doesn't give you much in this case. It's a blurry pattern that is copied over. But because you recognize the scene, as in one case being the street scene, in the other case being a dinner scene, you assign certain, so the other aspects come into your perception of the object. So basically that means that if you kind of take out the context, you could see that this doesn't really look like a dinner plate, or this doesn't really look like a car that much, maybe a little. But also their contrast is controlled for. So you want to have the contrast here match the contrast of its surrounding. It's the same pattern, but the contrast is matched to its surrounding. So the scene context is basically you're trying to use the information about the other objects appearing in the scene as uh, as cues for what one object could be. So this is another part of, so I'm calling these perceptual intelligence, meaning that it's not just, well, I'm looking at the visual input and recognizing it. No, you're looking at the visual input but you're looking at all the other stuff that comes with that visual input, all the context around it and everything else. You recognize that context, and then you recognize the object as well.
Show that it's distributed coding as opposed to specific coding. Does it have any relationship with this? Like
I don't think, yeah.
Different neurons to be able to recognize that object. It's not like specific to the object. It's we taking into account all the things surrounding. So that means that we're doing a distribution coding with different neurons. Is that what you're trying to show us, or
well,
that concept?
It is somewhat connected. Here, I wasn't trying to make that point. Here, I was just saying that when you have a visual pattern, what is it surrounded with impacts your perception of it, right? So if I look at this pattern, now it could be that you have a specificity coding that, so even if it's a specificity coding, right? It is getting input from other stuff. It cannot be, so meaning that at some point, it's getting a lot of input and it becomes more specific. And those inputs could be from the object itself, but it could be from the context around the object. So those context impacts your perception over time. It is distributed in a sense that you could say, well, the perception of the scene is also in your brain at that moment that you're deciding what the object is, and that impacts it. Yeah, I think there is connection there, but I don't know how clear that connection is, because a lot of time when we talk about object recognition and when models are designed for object recognition, they don't worry about the context too much, right? They try, the goal is that if I put this object on a blank screen without any context around it, you still want to be able to recognize it, because people can do that. Now, in this case, if I put this object on a blank screen, you may guess what it is. You could guess, I don't know, it's a car or a plate or, a mouse or something like that, a computer mouse. But a lot of those theories do not worry about the effect of context on that recognition performance. They're not too worried about that. But that's a good way to think about it, how your perception of the scene and the objects in that area, and that's, so we'll get to it in a little bit, but if I go back to this figure, just so because you're asking this question. Now, as you can see, these people put these sorts of identification and categorization. And again, what is the difference between categorization and identification? Well, the difference is that categorization is about saying, well, this is a dog. Identification is about what? Say, this is my dog, right? Or this is my friend's dog, right? So it's more specific than that. It's not just saying that, oh, this is a type of dog, or that this is a person, but rather this is my uncle, and this is his dog. So these sorts of, so that's what identification comes into it. It's not, it's a level above categorization. Now, these people aren't putting these things in the ventral pathway. The ventral pathway ends here at the area IT. Anterior IT to these people is the end of the ventral pathway. But these sorts of decisions about, okay, what the category, what is the name of this category? Or whether have I ever seen this category or this specific object before and identify it, those could happen somewhere else. And somewhere else, you could also have a scene graph. A scene graph meaning that, well, I'm looking at all of these people here and they're sitting in these chairs, so I have all this, you know what I mean? Like it's a higher level understanding of that scene. And that doesn't have to be in the IT necessarily. You could put all of that together in a higher area.
And would people's names be in either higher or different areas?
Yeah, it could be in the frontal or in the hippocampus type of area and those sorts of things. It's hard to pinpoint those things. But they're not in the IT. That's the goal. That's the point trying to make here, because you could, again, you gotta go back to some lesionist studies. If you lesion IT, then you're still able to, remember names and do all those sorts of things and whatnot. You still have an understanding of a scene graph or something, but you lose the ability to represent the features in a good enough way to recognize it, to learn new categories. Was there a question? Or?
With reading, we do also,
Hmm?
where does it happen in the brain?
Yeah, so there are.
Perception of words, we could like erase some letters in the word, it will still perceive the word, I mean, recognize.
Yeah, so
You know, where it happens.
yeah, so there are areas that people have mapped out for language processing, like Broca's areas or other areas. But a lot of that again has the recognition of words have to come in an early visual area, right? So a lot of again, this is somewhat the the progression of the course that we talk about recognition and visual processing, and you could apply a lot of these ideas to hearing and sound processing and word processing. We don't have time to get into that, but you could kind of apply the same intuition there, that you're processing these patterns, and then these patterns have to now get meanings at some point. At this point, we're not talking about that meaning that you assign to stuff. We're talking about, again, maybe in this course, we're not going to talk about meaning that you assign to stuff. At this point, we're really talking about that recognition part. Like if you're reading, what makes this word different visually from this word? Like that's the first step. You have to recognize that these are different letters of the alphabet that together are forming different words. So you could recognize them, and then you could put them together in terms of comprehension. So an experiment that people have done to talk about the effect of seeing contacts is some of these, again, these rapid presentation experiments where you could flash a scene, right, like this one, let people or show a scene, let people study this scene, and well, it looks like a kitchen counter with kitchen related stuff and cooking and bread related stuff, um, and then you could flash an image that is either scene consistent, in this case would be a white bread, or it's scene inconsistent but visually similar, like a mailbox, which is tastes like white bread, but and scene inconsistent target, right? Something that is visually similar, maybe visually dissimilar, and also scene this inconsistent. But then you could have these targets after you show the scene. You could have these targets flash very briefly. And what people could observe is that, well, people are more likely to detect the scene consistent object versus the other two options, right? So in a way, what could that mean? That means that you are more receptive, your pathways are more receptive to a scene consistent visual input, because you have these sorts of pre-process, right? If you're looking at this scene and you process this scene, right? The context, when you're perceiving the context of the scene, you're more likely to perceive something that is consistent with that scene. So you're more likely to look at a new object, and if it's consistent with that, you're more likely to recognize it because that's your expectation again. Now, in terms of the people have been thinking about this, and again, this is a model of top-down facilitation of visual recognition, right? So this is the pathway that we talked about a lot over the past couple of weeks, where you have a visual input, and this visual input can go through your ventral visual stream to the fusiform gyrus. Fusiform gyrus is part of the IT, so don't worry about that name. Like fusiform face area, for example, is in that. And you have early visual areas where the visual input comes through. However, you could have another pathway that is much slower. This is not as fast as the direct pathway that you have in the ventral, because which we saw examples of it when when you were able to quickly recognize these objects that are flashing one after another. That pathway is really fast. But you could also have a low spatial frequency. So a spatial frequency. So frequencies are, and again, in the context of a wave, you could imagine what high and low frequencies mean, right? In the context, so I talked about if you put your hand in water and just, move it so quickly, you have high frequency stuff. In an image, you could also have high frequency and low frequencies, and this is called low pass, high pass filter. Don't worry about it. But if you remove the high frequency stuff for an image, it looks blurry. When an image looks blurry, it lacks details, and those details would have been high frequency, but again, don't worry about it. This goes to the orbitofrontal cortex. So this is a frontal area, and that could impact the fusiform area. So in the case of this example, these are the same people who did this experiment. You could say, well, this perception of the scene, even though it's low resolution, even when you have a low resolution perception of the scene, you could create some understanding of the scene in your frontal area. And that understanding of the scene that is even low resolution can come down and change your perception of the object. Now, if the object is not ambiguous, this is not going to be, this will be a negligible effect. Because if the object is not ambiguous, you could look at it and recognize it. But if it's ambiguous, if it's hard to recognize, if it's occluded, then you're going to use that high level perception to kind of recognize it. Does that make sense? So the idea is that this may not be at play a lot of time, but that sometimes it becomes in play. When you want to use the scene context, for example, you could use this sorts of low resolution gist of the image to impact your processing in this pathway. So even in the example of, say, occlusion that we talked about, right? So you could recognize objects here. And again, you could recognize a lot of things that, well, you can imagine this is a relevant book and it's about cognitive or something. So you kind of use that top-down information to fill in the blanks, right? So you could kind of say, well, what could this be? It gotta be cognitive. What could the second word be? Well, it's gotta be psychology, right? So in a way, you're trying to fill in those blanks using top-down information. This idea of top-down and bottom-up, we'll talk a lot more about that after the second exam. And it's a very complex thing because a lot of time what is bottom-up and what is top-down is not clear. And what is feed-forward, what is feedback are similarly ambiguous concepts. But that's a language that a lot of people use, and you're going to encounter that a lot when you read papers as you go on in your careers and whatnot. But it's important. It's important in a sense that if something is top down, in this case, it's about, for a lot of people, is that it slows you down. And it only kicks in when it's needed. So if you have to use your understanding of the scene, then it slows you down. But what if you're using your priors of an object? Like, for example, in this example, well, you're using your priors. Is it a top-down thing? Is your knowledge of the physical world impacting your perception a top-down thing or a bottom-up thing? So it becomes really tricky. And again, it's a concept that you should be familiar with. It's not something that we're going to spend too much time on this exam, but I'm just giving you this is something that we will talk about when we go, especially the concept of attention. So a lot of attention modulations would come under top-down, right? If I'm attending to something, or if you're in a busy party and you're trying, like a cocktail party, and people are talking and you're trying to attend to someone, So in a way, you have all these top-down modulations in order to just hear one person's voice versus everyone else. So we're gonna reserve top-down for those sorts of processes. I think we're at the end of this. Any questions about anything we talked about today? Or stuff that we talked about the past few weeks? Yeah.
So what part of these slides will be on the exam or?
All of it. Yeah, this lecture is on the exam. The last time I was talking about, like earlier, I was talking about the new topic of attention will be after the exam, but everything in the past 4 weeks will be on the exam. I think so. I hope so. We'll see.